our discovery series through the NextGen seminar series. I'm Scott Rector. I'm a professor in the School of Medicine and also the director of the NextGen Precision Health Building. And it's my honor today to introduce Dr. Erika Boerman. A lot of you know her, and for those of you that do not know her, she's an assistant professor in medical pharmacology and physiology here in the School of Medicine at the University of Missouri. A little bit about Erika's background is that she did her doctoral training at Michigan State University, where she focused a lot on the calcium signaling and the regulation of vascular smooth muscle cell tone and function. She postdoced here at the University of Missouri, and she had some pretty cool innovative ideas during her postdoc, where she's able to kind of capture and monitor real-time vascular changes that are happening in the mesentery. So those vessels that kind of feed our gut. And then that's fed into her getting a pretty prestigious K99 R00 during that time, where she was examining these neurotransmitters that come in that can regulate vascular tone, regulate blood flow, and how they're linked to this condition called irritable bowel disease, inflammatory bowel disease, which is a chronic condition that's impacting millions of people. And we're struggling to find ways to manage that. She's now currently funded by an NIH R01. And what she's going to share with you today, and it's this local macrophages that are playing a role in the nervous stimulation in this condition of irritable bowel or inflammatory bowel disease. And so her presentation today is going to highlight that work and on this nerve-immune-adipose interactions and how blood flow changes in the mesenteric artery in this disease condition. So let's welcome Dr. Boerman, and I'm looking forward to an exciting talk.
Thank you very much for that nice introduction. And that was a great lead into what I'm doing now, because I think when people hear inflammatory bowel disease and know I'm a vascular physiologist, that doesn't necessarily make a lot of sense right away. So especially given that this is a broader audience, I wanted to kind of step back and take you through how I even ended up in this place and really convince you why this is important at all. So why do we even care about blood flow in IBD? We want to talk about what is this perivascular neuro-immune-adipose axis that I'm talking about here. That's not something that we just hear about all over the place. And I really want to address that in its three separate components today and talk about why each of these things is important in our work and in the disease itself. And then finally, I hope you come away with this understanding how these components function together in arteries with inflammatory bowel disease and some idea of how we're going to move forward to define these relationships. So starting with inflammatory bowel disease, I think a lot of people probably know somebody with Crohn's disease or ulcerative colitis, which are kind of the characteristic diseases that fall under that category. So you have Crohn's disease that can happen anywhere in the GI tract, whereas ulcerative colitis is focused primarily just in the large intestine. For how common this disease is and how much we've learned over time, we're still not sure why exactly people get IBD. So it is an autoimmune disease. And so there's some factors that it has in common with other autoimmune diseases. And we know certain categories that are related. So there's certainly a role for genetics, environmental factors, some of those being lifestyle factors, obviously immune system alterations. And then increasingly, we have an awareness that there's a microbiome component contributing to the etiology of that disease. And when you add that together, whether you have mild disease or severe disease, kind of the highlighting factor is that you have a chronic aberrant immune response and chronic inflammation, both within and outside the intestine. And one of the reasons we care about this is a disease that's on the rise. Right now, we're at about 1.3% of the US population, and we have more IBD cases per capita than just about anybody in the world. But if you look worldwide, we're still on a rapid increasing trajectory. So the US is still coming out of this phase here where you have an increase in both incidence and prevalence. And we're getting to the part where we're no longer having as much increase in incidence, and that's flattening out a little bit. But the rest of the world is very much still in this rapid rising phase. So we're going to continue seeing this be a huge problem worldwide, especially as more developing countries shift into this rapid rise phase. And what we want to see is the second half of this graph, which is to see things peak and start to come down. And for that to happen, we need better treatments. We need to increase, more effective cost-saving treatments, personalized therapy, and primary prevention. So this is one of the reasons I think it's important to look places outside the gut to tackle these symptoms in this disease. So I'm a vascular physiologist, so it makes sense that I would come to this from a vascular angle. But this is not just an idea that came out of my mind. There's a strong link between IBD and cardiovascular diseases. So if you have inflammatory bowel disease, you're significantly more likely to have ischemic heart disease, cerebrovascular disease, things like stroke, heart failure, atrial fibrillation, hypertension, diabetes, pretty much any cardiovascular disease, you're at increased risk for cardiovascular disease. And one thing that's interesting here is that this is not a population that's overweight. It's not a population that tends to have diabetes or hypertension in huge amounts. So these are a population that tends to have low traditional risk factors, and yet they're having a higher incidence and risk for these diseases. And the other thing that made me really interested in getting into this area is that people with IBD have documented decreases in intestinal blood flow. And this varies by location and disease severity, but it's common enough that looking at this can actually be diagnostic for the disease in the early stages. So when I started doing this work, we wanted to know just what happens with vascular function in IBD. There surprisingly was very little known outside of some basic blood flow studies. So starting from the first part of my Neuro-Immune-Adipose axis, I want to talk a little bit about the first place we started is perivascular nerve function, or in this case, dysfunction in IBD. So if you're not familiar with perivascular nerves, if you look at the resistance vasculature, almost all of the arteries are significantly innervated with both sympathetic and sensory nerves. And this image shows a good example of that. If you look at the outer layer of the blood vessel, which we call the adventitia, it's very much covered in perivascular nerves, almost like if you wrapped chicken wire around the vessel, it kind of looks like that. In this example, you see a vessel stained for PGP 9.5, which stands for all perivascular nerves. And there's two main types that we're interested in here. There's sympathetic nerves that I think most of you are probably familiar with. These are your vasoconstriction, fight or flight, noradrenaline nerves. And then you have sensory nerves that kind of do the opposite. These are going to relax your blood vessels. They're going to increase blood flow, so they're vasodilatory. And today we're going to focus primarily on these sensory vasodilation nerves, because if you think about somebody with IBD who's having restricted blood flow to the gut, it makes sense that maybe they're having a problem with this vasodilatory pathway. So that's what I'm going to focus on in some of our work in this area. And if you look at these sensory nerves, in the schematic that shows kind of the cross section of the vessel wall, you have your endothelial cells that touch the blood, followed by smooth muscle cell layer, and then the nerves up above in the adventitia. Sensory nerves release two vasodilatory neurotransmitters, calcitonin gene-related peptide, or CGRP, and substance P, or SP. And when they're released, they bind to their receptors on the smooth muscle and or endothelial cells. And then ultimately downstream, you get sensory vasodilation. And a lot of what we study is looking at how different things change the ability of these nerves to cause sensory vasodilation. And we already know that these nerves are dysfunctional in some conditions. So a lot of cardiovascular diseases, diabetes, hypertension, obesity, Alzheimer's, and from my particular background of aging, we know that there's a lot of conditions in which you see dysfunction in these nerves. One of the reasons I was particularly interested in this is that in addition to causing vasodilation, CGRP and substance P have other effects in the body outside of the vasculature. So they can also modulate things like pain, GI motility, immune responses throughout the body, and inflammation. And so when I started with this project, it was kind of my theory that maybe an imbalance in CGRP and substance P was an important role in what we saw with vascular problems in IBD. So we decided to kind of go down that road. And again, we focused on CGRP and substance P. And in humans, if you look at serum levels of these two things, you tend to see a decrease in CGRP, which is more anti-inflammatory, and an increase in substance P, which is more pro-inflammatory. And these differences increase and decrease respectively if you consider the severity of their disease. So how do we study this? We don't do any human studies right now. So everything we do is in mouse models of IBD. And right now our model is the IL-10 knockout plus Helicobacter hepaticus model of IBD. So IL-10 is a really important anti-inflammatory cytokine. And if you knock that out of the mice, you kind of prime them for this inflammation, but they don't get IBD on their own. But once you introduce this commensal gut bacteria, Helicobacter hepaticus, which we do this twice after weaning via oral gavage, then they start to develop IBD. And after this Helicobacter gavage, we allow them to develop disease for 90 days. And then we start doing our experiments. And as you can see after this time point, if you look in the gut where the disease is more evident, you can see in control versus IBD mice a significant amount of inflammation and hyperplasia that happens in that colon epithelium. And pretty much everything that we do uses first and second order mesenteric arteries. So these are branches off the superior mesenteric artery here. There's a first order vessel. The branches off that are the second order vessels. And we primarily use vessels in the ileum, which is the terminal part of the small intestine before the large intestine. And then sometimes going back as far as the terminal jejunum. So that's what we're using for everything I'm going to talk about today. So if we start looking at some isolated vessel studies, one of the major techniques that we use to study blood vessel function and perivascular nerve function is called pressure myography, where we can isolate a mesenteric artery like this and cannulate it onto 2 glass pipettes and then warm it, superfuse it with saline solution, and you can study it as though it's still in the body. And then with electrical field stimulation, we can look at the function of those perivascular nerves. And to study sensory nerves specifically, we have kind of a 3-step protocol we use. So we pre-constrict those arteries with phenylephrine, then we block the vasoconstrictor nerves with a compound called guanethidine or GE here. And then when you electrically stimulate the nerves, you can look at vasodilation as we see here, and that's sensory vasodilation. And when we did this in our control and IBD mice, you can see control in the black and IBD in the gold. We're looking at 2 frequencies of stimulation, 8 and 16 hertz, and then the maximum dilation, percent max dilation on the y-axis. In control mice, you see a nice frequency dependent vasodilation that's very significant, goes up as high as 80% in some of these mice. By contrast, if you look in the IBD mice, this is severely impaired at both frequencies. This is a very significant reduction. So these vessels just aren't really able to do sensory vasodilation in the normal way. And we're not showing the data here, but another important function of sensory nerves is that it inhibits sympathetic vasoconstriction. And in other experiments, we showed that they also lose the ability to inhibit that sympathetic vasoconstriction. So this was kind of a key piece of data for us that said, yes, there is sensory dysfunction. There's a lack of vasodilation in these vessels. So something is going wrong here. To follow up on that, we did a few other experiments. I'm going to go through this relatively quickly because this data is all published. So if you're asking, why is there something wrong with sensory dilation, there's a few avenues you can go down. The first thing we wanted to do is look at the density of the nerves themselves. And so we labeled vessels for CGRP and substance P, and tyrosine hydroxylase for sympathetic nerves, and then we quantified that data. And there were some differences, a slight increase in substance P nerves, a slight decrease in sympathetic nerves, which is actually the opposite of what we expected to see, but nothing really dramatic that probably explains that significant loss of sensory vasodilation. The same was true when we looked at the content and release of those neurotransmitters. So if you grind up these mesenteric arteries and just measure the amount of CGRP and substance P in them, you have a trend that's not significant towards decreased content for both CGRP and substance P. And if we measure the amount of transmitter that's released in the bath when you electrically stimulate these vessels, you see the same phenomenon. A non-significant trend towards decreased release of both CGRP and substance P. So the same thing, this could certainly contribute, but it's not major like what we would expect. So next we looked at the receptor function and expression of the receptors for these CGRP and substance P receptors. And we did that by doing concentration response curves to each neurotransmitter where we're providing the neurotransmitter. And when you do that with CGRP, looking at increase in concentration on the X and max dilation on the Y, you can see there's really no difference between control and IBD. In contrast, we see a pretty big reduction in the ability to vasodilate to substance P specifically. So this implies some sort of receptor dysfunction in the substance P receptor or neurokinin 1 receptor. And then when we label for the actual receptors and image that confocally, we found kind of a corresponding answer. So there's robust staining for RAMP1, which is a CGRP receptor protein in both control and IBD, but not much difference in smooth muscle or shown here endothelial cells. In contrast, NK1, we saw a big difference, but what we actually saw was an increase from control to IBD in endothelial NK1 staining. So these receptors are there, they're actually there in a bigger amount, but it seems like something about it is non-functional. And so this kind of leaves us, we've seen a number of things that are trending, they're sort of significant. There's some differences in the density of these nerves and the content release of the transmitters. It seems like there's some difference in the expression and function of the receptors, but it's unclear if these things are cumulatively enough to really cause those big differences that we saw before. So we decided to pursue some other avenues. And that leads into the next part of our axis, which is the adventitial macrophages in IBD. And I think this seems like it came out of nowhere. And for us, it kind of did, because one of the ways we came upon this avenue of experimentation or really even thinking is that we weren't really sure where to proceed next. And so one of the things that we did is an RNA sequencing of these intact arteries, a bulk RNA sequencing in control in IBD mice. And when we looked at what was different, there was about 200 genes that were differentially expressed between control and IBD. And we found that there was a significant increase in various macrophage markers. So these were all in the top 30. We see a lot of different macrophage markers, CD163, MIP, CD68, MAC1/CD11B, F480, and down the list. So this seemed like something that was significant, and we decided to pursue this area a little bit further. And we wanted to know, do these macrophages accumulate in the adventitia with IBD? So one thing that I didn't show in that previous cross section showing the different parts of the blood vessel is that in addition to having a lot of perivascular nerves, the adventitia also has a lot of resident immune cells. This is something that's known, it's healthy, and it is possible for these immune cell populations to change in different disease states. This has been defined in a number of different diseases, and a number of investigators have looked at this in really elegant ways in obesity and diabetes and other conditions. So this is something we wanted to look at in IBD as well. And then second, assuming that we do see changes in this population of macrophages, what happens if you get rid of them? Can you actually change nerve function? And the way we did that is with claudronate liposomes. So if you're not familiar with this technique, you inject the mice IP with liposomes that contain either saline for the sham group or claudronate. And basically what happens is the macrophages will engulf that claudronate liposome, the claudronate is released and triggers apoptosis in the macrophage. And this little animation actually came from the company that makes those liposomes. So we'll call this a scientific explanation of what happens. And then after that, we're proceeding essentially on the same timeline we're on before. So they developed disease for 90 days, received their injections of either PBS or claudronate liposomes, and that left us with 4 groups that we're studying. For both control and IBD, we have PBS group and a claudronate group. So moving forward, that's the groups that we're looking at. So then we just repeated the experiments that we were looking at before at sensory vasodilation. So here, instead of showing just 8 and 16 hertz, I'm showing the full range of stimulation frequencies, but the experiment is the same. And so if you look at control in the black, you can see a nice frequency-dependent vasodilation that's significantly inhibited in IBD, which is exactly what we saw the first time. This is what happens in the PBS liposome group. You see decreased sensory vasodilation with IBD. When we repeat that in the mice that have the claudronate liposomes, nothing happened in the control group. So if you look at black dashed line to black solid line, depleting macrophages didn't have any significant effect in that group. However, when we looked at the IBD group, you can see this huge increase in vasodilation, sensory vasodilation, shifting from the solid line here to the dashed line above. That happens when you deplete these macrophages. And again, we're not showing this here, but it also improved the ability of sensory nerves to oppose sympathetic vasoconstriction, which kind of increases vasodilation in a roundabout way. So this was a really important finding, showing that it seems like there are some important roles functionally in these vessels for these macrophages. And what we found, I'm not going to go through all the staining that we showed here because it takes a long time to go through all that labeling, but if you label these vessels, you can see a huge increase in CD68, CD163, antigen presenting cells, MHC2, and just total leukocytes or CD45 positive staining. And those accumulate in the adventitia with IBD. And Macrophage depletion with claudronate restores that IBD impaired sensory vasodilation without altering perivascular nerve density very significantly. So this was a really important finding for this. And this was really the basis when I wrote my R01 grant on investigating this macrophage interaction a little bit more deeply. And so the third part of our axis that I want to talk about, and probably the newest data that we're working on right now, has to do with the perivascular adipose tissue, or PVAT, in IBD. So when I showed the first schematic, I left out a really key part of the vasculature. So normally we talk about 3 layers of the blood vessel: the endothelial layer, smooth muscle layer, and adventitia. But really the perivascular adipose is increasingly understood as an extremely important regulator of vascular function, probably for about 20 years now. Since the early 2000s, we've seen a huge increase in the amount of work showing that this fat is not just here. Even when I trained in the early 2000s, the only thing we ever did with perivascular fat is cut it off so that we could do vascular function experiments. So now we're starting to get a more nuanced appreciation that this adipose, and really all adipose in the body, is truly an endocrine organ that's affecting vascular function and can have significant effects. And also important for our sensory nerve experiments, the adventitia that's innervated with these sensory nerves, those are nerves continuously protrude into the PVAT. So this is not 2 separate layers of innervation, it's one continuous layer of innervation. So it is important that we started looking at that as one continuous layer. And PVAT in general, if you look at a healthy vasculature, is what we call anti-contractile. So it tends to help vessels stay dilated, stay relaxed, but it actually releases a whole multitude of different factors. So within this adipose, you have a lot of different things other than just adipocytes. You have a number of immune cell populations. You can get release of exosomes, microvesicles, and a number of other things. Some of these are what we call adipocytokines, which are just cytokines released from the adipocytes. And then it also releases vasoactive factors, both relaxing and constricting. And the net effect of this is usually vasodilation, but this is something that can easily change depending on the local and global environment that they're sitting in. And so this is something we wanted to look at. And we wanted to know, how does IBD affect the PVAT adipocyte properties? So not just the whole net effect of what happens, but what's going on with these adipocytes themselves in the PVAT. We wanted to know how the PVAT macrophage population changes during IBD pathogenesis. We saw a huge change in the macrophage population in the adventitia wall, so it stood to reason that a similar thing might be happening in the PVAT. And then finally, can modulation of the PVAT macrophage population alter vascular function, similarly to what we saw in the vascular adventitia? So when we looked at the adipocytes themselves, what we did is just take cross sections of PVAT tissue surrounding mesenteric arteries and labeled with hematoxylin and eosin. And I hope you can appreciate the difference just looking at the control versus IBD, that there's a lot more variation in the cell size. The cells tend to be a little bit smaller, and there's a little bit more of what we call the stromal vascular fraction, which is basically all the other stuff that's in the adipose tissue other than the adipocytes. And when we quantify that, this is what we saw. So we did see a big decrease in the average size of the adipocytes and a corresponding increase in the number of cells per field of view. So we weren't seeing less total cells, just an expansion of small cells. And this happens in conjunction with a decrease in body weight and no change in the mass of the PVAT itself. So one reason it's important to look at some of these things is that a lot of what we know about PVAT is garnered from diabetes and obesity research, where you see big hypertrophy of these mesenteric adipocytes. And that's just not usually what we see in this population. That's usually losing weight as their disease gets more serious. And that isn't what we saw. So we actually saw some hyperplasia of smaller adipocytes, and that's interesting. So we haven't quantified the stromal vascular fraction, but looking at it, it does seem like there is a little bit more there, and I think that's something we'll go back and do soon. So knowing that, we wanted to move on and say, do M1 and M2 macrophage populations change in PVAT during IBD pathogenesis? So when we're looking at our macrophage populations, we want to use 2 different populations because mostly they're defined on kind of 2 classically defined, at least from cell culture populations. You have M1 macrophages that are considered pro-inflammatory, and M2 macrophages, which are either considered anti-inflammatory or have a more complex phenotype that's difficult to define. And even though these things occur on a spectrum, we did use a couple different specific markers to look at this. And most importantly, instead of just looking at our 90-day post-gavage time point, we wanted to look at these macrophage changes throughout the development of IBD. And so we selected time points starting from day 0, which is before gavage, all the way out to day 240 post-gavage. And one of the reasons we did this is because that spans about the equivalent of 30 human years. And most IBD patients are diagnosed between ages 15 and 30. So we felt like in terms of translation, this encompasses a lot of the ages in which people are really ramping up in their progression of IBD. So I'm not going to show too many images here, but just as an example of day 240, we just did confocal imaging as before, looking at our different macrophage labels, F480 for total macrophages, CD206 for our M2 or maybe more anti-inflammatory macrophages. And then we used CD68 as our M1-like marker. And then this is just a merge of all the images. And we quantified the percent area of the PVAT that's occupied by each of these labels. And you can see, at least in this example, that looking from control to IBD, it seems like we have an increase. If you look green to green, red to red, magenta to magenta, it seems like there's an increase in all of these markers. But fortunately, I had a master's student, Sam Jenkins, who went through and did all of these time points, all of the imaging and all of the analysis so that we can look at these summary graphs now. And if you look at F480, which is total macrophages over time, in black, you can see that for about the first 20 days, you have a sharp increase in total macrophages that kind of plateaus out after that point. And this actually is not, we thought it would just stay flat over time. And kind of the opposite happens in IBD. It actually starts to decrease for those first 20 days, stays a little bit flat, and then trends up a little bit at the end. And I think that one thing we didn't consider in these early time points is just the effect of normal development. So in addition to developing disease, especially in the control group where you don't have IBD, you just have normal growth and development during that time and probably a natural flux in the population of macrophages. If you look at our M2-like macrophages or our anti-inflammatory macrophages, you can see that the two populations kind of crisscross over time. So in the controls, they rise and then hold steady, whereas you see kind of a consistent decrease over time in the IBD group, suggesting a loss of those anti-inflammatory macrophages. The opposite was true in CD68 or more M1-like macrophages. Pretty much this is what we thought would happen everywhere, a more flat population in controls over time, and we did see that here. But in the other group, in the IBDs, we see that sharp decrease that hangs in there till about day 40, and then a rapid increase. And one of the other ways that you can look at this that better describes kind of the overall picture is to look at the ratio of the M1 and M2-like macrophages. So this is a CD206, CD68 ratio or an M2 to M1 ratio. And I hope you can appreciate how much these two lines diverge over time. So it does seem like it happens pretty early on and then just stays separate as the disease progresses. So one of the things we were hoping we would see is a huge change right away, and then maybe lesser happening right away, lesser happening as time progressed, suggesting that maybe you could do a really early intervention here. But this data does not show that so clearly, but it does suggest that changes in this macrophage population are extremely evident in IBD and represent a promising avenue to pursue. And one of the other things that looking at this PVAT data in general that we're really excited about is that it seems like PVAT shares properties with what they call creeping fat. So if you look at Crohn's disease patients, they have this characteristic buildup of fat around the intestines and mesentery called creeping fat. And it's not like what you see with visceral fat in obesity, where you have hypertrophy of each adipocyte, an increase in M1 specific macrophages. What you see in creeping fat and what we saw here is adipocyte hyperplasia without hypertrophy. So you see an increase in the number, but not an increase in the size of those adipocytes. We see perivascular inflammation. we do see an expansion of the macrophage population and persistence of M2s within that population. So we're not looking at just a switch of the same population from M1 to M2 like. We're seeing an expansion of the whole population with both types still very well represented. And then finally, our H&E staining did suggest a little bit of adipose fibrosis, but this is more descriptive right now until we do some further experiments. So where does that leave us with this Neuro-Immune-Adipose axis? So we see this big influx of macrophages, both in the PVAT and in the adventitia. And so we wanted to get rid of them and see what happened to vascular function with and without PVAT and with and without macrophages. So we repeated our clodronate experiment and then tested the function of perivascular nerves in vessels with and without PVAT. And this is just the same protocol I showed before, looking at induction of disease, waiting for disease to develop, and then treating with clodronate. So one of the most important things that we found without even looking at the clodronate liposomes is that similar to what you see in other disease states, PVAT does become procontractile with IBD. So this graph on the left is showing just control vessels. And you can see the dashed line shows vessels without adipose compared to the solid line with adipose. And this is again, just a frequency dependent constriction, looking at sympathetic vasoconstriction. And I hope you can appreciate that when you go from no fat to with fat, you see a decrease in that vasoconstriction. And the difference between these lines shows you how anti-contractile that PVAT is. When we repeat that in the IBD vessels, we see the exact opposite happening. And again, these aren't treated with clodronate. These all got the PBS liposome. So if you look at the vessels with no fat versus the ones with fat, you actually see an increase in the amount of vasoconstriction. And this is the exact opposite of how healthy PVAT should be functioning. So we definitely have some sick PVAT on our hands here. And this is a phenomenon that has been defined again in other diseases. So I'm going to show you just the data for IBD for the clodronate, because otherwise we're going to have 8 lines instead of 4 lines. But I will tell you that clodronate did not do anything noteworthy in the controls. So nothing changed with the addition of clodronate. So they were PVAT was anti-contractile. It remained anti-contractile after macrophage depletion. In contrast, when you repeat the experiment in the looking at the anti-contractility or pro-contractility in PVAT after macrophage depletion, you see a huge difference. So down here at the bottom, you can see if you go black to black line, you can see a huge change from the difference in PBS versus clodronate liposomes. And again, we didn't see any difference in vessels that didn't have the fat. But if you have the fat present, depleting macrophages causes essentially elimination of that procontractile phenotype that we saw before. So in vessels that have PVAT and PBS liposomes, so with macrophages, still procontractile. After macrophage depletion without fat, there isn't an effect. But if you leave the fat on and look at the ones with no macrophages, you decrease that PVAT procontractility specifically. So similar to what we saw in the vessel adventitia, it seems like getting rid of this macrophage population, whether it's M2s or M1s, seems to be doing good things in terms of vascular function. And I should mention that clodronate is not a nuanced approach. This is depleting macrophages everywhere in the body, not just the vasculature. So we're really hitting it with the sledgehammer here. So everything is gone in these, not just in the vasculature. So when we repeat our sensory nerve experiments, we know it's changing the contractile ability of PVAT into making it anti-contractile again, but what is it doing in terms of sensory nerve function? Because this is one of the areas we're most interested in. So here I'm showing just one frequency, 16 Hertz, which is our max frequency to make it a little easier to look at. And as you can see in control, this is just PBS from minus fat to plus fat. You see that sensory vasodilation is better when you leave that PVAT on. So maybe sensory nerves have something to do with that anti-contractile function in healthy, non-diseased PVAT. So this PVAT is increasing sensory vasodilation. In contrast, when we look at the PBS group in the IBD same as before. We saw very limited sensory vasodilation. Going back to the clodronate studies, if we look again on the control side, you can see minus fat to minus fat, getting rid of macrophages actually improves the sensory vasodilation. But from with fat to with fat, there's really no difference. So clodronate increased sensory vasodilation, but only in vessels without fat. And that is not what we expected. So it seems like in the controls that PVAT, but not its macrophages, are important for sensory vasodilation. That's a little bit more complicated than we were hoping the answer would be, but science rarely gives you those straightforward answers. And then looking at the IBD group, I hope you can appreciate that in the minus fat group, you get a huge increase in sensory vasodilation. When you repeat in the vessels that have fat, you get an increase in sensory vasodilation, but not to the same extent as we see in the vessels without fat. So in the IBDs, macrophage depletion increases sensory dilation, whether or not you have perivascular adipose, but it actually does a little bit better if you don't have it. And we're still kind of wrapping our head around what exactly we think that means physiologically. So just to summarize everything that I've talked about so far in this axis, in a healthy animal, this is kind of what we have going on. So you have your sympathetic nerves and sensory nerves that can cause constriction and dilation respectively. And you have a number of things that act as checks and balances on this sympathetic constriction. You have sensory nerves inhibiting that. You have the actions of PVAT inhibiting that. And the net effect of that is normal GI blood flow. Based on our experiments, it seems like a lot of factors here change. So sensory nerves are losing their ability to inhibit sympathetic vasoconstriction. PVAT is losing its ability to inhibit sympathetic vasoconstriction. And a lot of these changes seem to be very related to a change in the macrophage population, both in the adventitia and in the perivascular adipose. And likely the net effect of this is what we see in human patients, which is decreased blood flow. So since we have a little bit of time left, I want to talk about where we're going to go from here. So we've defined some really interesting changes, but we don't really know how these things are changing or a lot of mechanism behind what we're looking at. So we really want to know, how do these macrophages interfere with perivascular nerve function? And one thing that I just continuously come back to is the extremely close proximity of macrophages and nerves. So this is actually a vessel that's stained in red for perivascular nerves, and green is not macrophages, it's CD45, so any leukocytes. But I hope you can appreciate, if you look at this red nerve here, how closely entwined that these immune cells are with the nerves. They're literally on top of them, going in and out of them. It's almost like they're one. And this is not some kind of staining aberration. So if we look at this with what we call matte green mice or CSF1R GFP mice that have fluorescently labeled macrophages, you can still see staining showing up that looks exactly like perivascular nerves. And that's because these projections from the nerves really track with these perivascular nerves. And again, this is not even this is not an IBD mouse. This is just a healthy animal, a mesenteric artery. And we see the same thing happening in the PVAT, as you can see here. Another thing we want to look at is cytokine and adipokine release profiles. So this is really difficult to do using just isolated single vessels. Not enough is released to really measure from the bath solution. So Cassandra Ramos in the lab, who's in here today, is going to take on a vessel culture project where we can do some explant culture of both fat and vessels, either separately or together, and see what kind of cytokines and adipokines are released both under control conditions and after stimulation with sensory neurotransmitters, and how that differs in control and IBD. So this will be like a huge change for me doing physiology into like more of a culture situation. So that will be really interesting for the lab. And then also we want to look at macrophage CGRP and substance P receptor expression and signaling. One of the theories that I cannot get out of my head is that I feel like one of the reasons we lose sensory neurotransmitter induced vasodilation is that you have CGRP and substance P released from those nerves. And instead of diffusing down and acting on smooth muscle cells and endothelial cells, they're binding to receptors that are on the much, much closer macrophages right next to those vessels. And then instead of causing vasodilation downstream, they're causing immune changes and possibly inflammation. through mechanisms involved with these nearby macrophages. And just in the past couple of months, I had a very talented PhD student rotate through my lab, Olu Latif, and he did some imaging of macrophages labeled here for F480, as well as the CGRP and substance P receptors, RAMP1 for CGRP receptor and NK1, which are substance P receptors. And if you look at this merged image, you can see that there's a huge amount of overlap between this. First, you can see that the nerves, there is no nerve staining here. So any nerves that are evident, which they are very evident, are there because they're labeled for other things. And you can see that these nerves do express receptors for their own transmitter, which we knew. But there's also a lot of staining in these macrophages. So we do want to go and do some more mechanistic studies, which will probably take us a step back from in vivo physiology, but could give us some really important answers for whether these transmitters are binding and what happens when they do in healthy and IBD mice. We've also repeated our RNA sequencing in PFAT because that gave us some really interesting answers before. And this is really preliminary. We haven't done much in-depth analysis with this data yet, but if you look at the things that are increased in PVAT with IBD, everything is related to inflammation, immune responses, and what seems to me a lot of things that might be related to the NLRP3 inflammasome, things like IL-1 beta, interferons, pattern recognition pathway, positive regulation of TNF. So these are all things that are kind of highlight features of the NLRP3 inflammasome, and that's something that we'd like to look at further. And one thing that was interesting, which seems obvious that now that I look at it here, is that if what's decreased the most in PVAT are mostly metabolic things. So there's a huge change in metabolic function of these adipocytes in IBD. And again, in retrospect, of course it is, because adipose is an extremely functional endocrine organ that's doing a lot. And so I think that changes in these pathways are obviously extremely related to the changes in immune function above. And that's something that I'm personally going to have to learn a little bit more about before we pursue that angle. We also want to know if the nerves and the fat itself drives that macrophage infiltration. So what actually causes these macrophages to exist in higher numbers? Is it proliferation of cells that are already there? Is it cells that are coming in from the outside? We're not sure. And so to see if the nerves themselves are driving that influx, we can do sensory denervation. And we've done some pilot studies on this. Fortunately, these sensory perivascular nerves form after birth. So if you give them an injection of capsaicin within 1 day of birth, then you can prevent the formation of these perivascular sensory nerves. And it's been a little complicated because you have to get the mother to reaccept the pups after you've done this capsaicin injection, which has been a moving target, but we're working on that. So that is all I have. I wanted to leave some time for discussion, but I have to thank everybody that's helped me with this. Liz Gruns is my lab manager. None of this would, if she ever quits, I'll just quit. A number of talented students who have come through the lab, including Olu, who just did a rotation and generated some data. The informatics core, who has helped me a lot with getting that data analyzed for somebody who is not accustomed to handling that amount of data. And then just from my career trajectory from postdoc till now, my many, many official and unofficial advisors here. So too many to name at once, but I would like to thank all of those people. Obviously, the Medical Pharmacology and Physiology Department, NIH for the funding. I had to include my two favorite work at home colleagues here, the cats. And then as of yesterday, I have, if you're a college basketball fan, I have to thank my alma mater, Michigan State, for hanging in there.
Thanks a lot, Dr. Boerman.
Yep.
We have time for questions, about 10 minutes or so. I'm going to hand a microphone so that those on Zoom can hear as well.
Thank you. That was very interesting. Most of the treatment we have for IBD involves attacking the immune system, activated T cells and such. Do you have any idea whether any of these abnormalities you presented today may lend themselves to being treated for IBD?
Yeah, so there's been just a few studies looking at blood flow in humans after treatment with things like Humira and other immune modulators. And there does seem to be improvement there, but there's never been any real nuanced studies. I think that if you look at the wheel of whether vascular problems are a cause or an effect of IBD, I think there's certainly more of an effect. So anything that is harnessing change in whole body inflammation is going to have a positive effect on the vasculature as well. But one of our biggest problems with those immune modulating drugs, especially as the IBD population is getting older, both in just time with the disease, and we now have this big population of older onset, those patients do not tolerate those drugs well. And so we need other treatments that we can give to people who can't tolerate these drugs. So I do think they're very effective, and I think that they could have some important effects on this endpoint in terms of vascular function, but I think it's important to maybe try and develop some treatments that aren't related to those.
Yeah, yes, the very good talk. So we are also interested in the macrophage. The IBD you show the data is clearly increase the macrophage in IBD. So do you know, so actually, so usually the macrophage is from the two sources. One is resident, one is monocyte derived, right? So does your the data show the increase the macrophage is from the resident tissue is proliferation, or is from the monocyte induced differentiation?
Yeah, so the answer is I don't know. And I think that's an extremely important question to answer. I think that the particular area we're looking at would be a really good case for what I call outside-in inflammation, just peritoneal macrophages coming into that PVET that's right nearby, and then subsequently into the vascular adventitia. But I certainly think that monocyte-derived macrophages are part of it, both ones that come through the endothelium in the bloodstream and an expansion of the population that's already resident in both the adventitia and the PVET. So I don't know the answer, but I do think that's an important thing to follow up on.
Okay, I have a second question. So you also show is adipocytes is changing there, right? Macrophage phenotype change also is there. Do you think so is the adipocytes change driven the macrophage change, or is the macrophage change driven the adipocytes change there?
I think that the adipocytes might be a major driver of that. And I didn't show this, but Cassandra did the same time point study looking just at the adventitia over time, and the changes are a lot more modest. And so it seems like the PVET is probably the major driver of that, which makes sense. It's a bigger tissue, bigger macrophage population. And so I think that changes in the adipocytes themselves probably have a lot to do with that. You know, as I said, as you get metabolic changes, then you get changes in the adipocytokines that are released, and then ultimately changing the combination of vasoactive factors that are released as well. So that's something when we get into the culture model that we really want to try and quantify some of those players.
Great talk. So is this IBD model a standard model, ALTEN knockout plus hepaticus?
So there's a number of models that people use, mouse models of IBD, and I would consider this one of the three major models that people use. So IL-10 knockout is one of the major models. If you use a C3 background mouse, then you don't need to treat it with anything. They just have IBD. But IL-10s, you need some sort of stimulus. A number of people use Helicobacter hepaticus, but different people use different things to trigger the disease. So some people use E. coli, some people use LPS, some people will give DSS or chemical irritant. And then I would say the other arm is what they call T-cell transfer models, where you do adaptive T-cell transfer to induce IBD. And that's also an extremely rigorous and valid, commonly used model, but this is one of the standard models.
So your controls are just the hepaticus or the IL-10 knockout mice, the control for IBD.
So they're both IL-10 knockouts. In most of what I've shown today, both groups are IL-10 knockout, and it's just whether or not they've received the gavage. We have done a lot of experiments where we also used C57s as controls. We haven't found any major differences in vascular phenotypes between the non-gavaged IL-10s and the C57s. And over time, we have found it difficult to control the phenotype of the non-gavaged IL-10s. So throughout this period of time, this colony has had to move 5 times in 5 years from different facilities. And we have had a lot of trouble recapitulating that control phenomenon. So we've mostly moved to using C57s as our control. And I think I will probably stick with that. But like for the RNA sequencing, we included like wild types, IL-10 knockout and gavage IL-10
I see,
knockout.
because I wonder in the ALT knockout mice, since they are already immunosuppressed, right? They might have increased macrophages population.
Yes,
Do they
that-
have in a basal label?
Well, I mean, the ones I showed before, the controls were also IL-10 knockouts. So in that example, you don't see any IL-10 mediated increase. And so we have not tested that rigorously where we actually went through and counted, but we have not seen significant differences between a non-gavaged IL-10 and a C57 in terms of the PVAT and adventitial macrophage.
So in the
population.
IBD patients, is IL-10 less? Have you measured cytokines in this mouse, just in the plasma? Other inflammatory cytokines?
So we've done some cytokine assays like with PCR, and certainly the RNA sequencing confirmed that there was a huge difference in IL-10, of course, because it can't make it. So yeah, there's a big difference in IL-10, but because of that, when we did the RNA sequencing, we made sure to include both IL-10 knockout groups and the C57 group. But I certainly think that that is a valid consideration that, of course, the IL-10 knockout is causing whole body immune changes on its own.
Thank you for the very interesting talk. One of the things that macrophages do, and perhaps also the adipocytes, is to secrete proteases, proteolytic enzymes. And certainly proteolytic enzymes overlap many of the processes that you listed on the IBD list as being upregulated. And so, and I also wonder about the possibility of proteolytic enzymes damaging some of these receptors that you referred to early in your talk.
Yeah.
And so, you know, do you have any comment? Is that on your radar screen?
So we haven't thought a lot about that, but I actually did have a student in the lab who did an entire project looking at endothelin converting enzyme and neprilysin. Not so much from the protease angle, but from changes in the receptor trafficking. And we do find significant differences in those two things, ECE1 and neprilysin, between control and IBD in the vasculature and in the serum. So in terms of those two things, yes, but that's not something that we've really thought about. But I think that would be really interesting because that speaks to a lot of other things in the vessel wall, like stiffness and things like that as well.
Great talk. So from the immunological perspective, when I look at it, and then this is since, you know, inflammatory based disease, have you looked at it, Th17 cells, especially IL-23 secreting Th17 cells in your model?
We have not done that. And I would actually consider that probably one of the primary weaknesses in what we've done so far is that we focused exclusively on macrophages when clearly there's going to be differences in other immune cell populations. So right now we're just limited by the amount of people and time that we have in the lab to go down these different avenues. But I certainly think that looking at, you know, T cells, neutrophils, mast cells, all of those things would be really important to look at moving forward.
I had a quick question. How far are we from translational studies?
Like the big we or me personally?
How about you, but the big we? What about us as a collective?
So capital we, I think that we could certainly do more. I think that if we had a bigger IBD population being seen at this university, something looking at blood flow changes in that population, even based on the number of treatments they are and aren't having now, would be a really interesting, rigorous way to look at this. We've done some of our most important findings. We've validated with imaging by getting samples from the biorepository and making sure we see changes in some of these important markers in the human vessels as well. And I should say we can't get mesenteric artery samples because that's not something that commonly comes out surgically. But if you get bowel resection samples, the biorepository is great and will give you samples that include vasculature. And so we do see some of these changes recapitulated. So if that counts as translation, we're doing some translational work, but I have not in my career done any human subjects work. So I don't want to tell you that we're really close to doing that because I don't think that's reality, but I would love to see more of that happening. And I think that with the expansion of our population in the School of Medicine researchers here, it's definitely possible.
All right, so you opened the door twice. You mentioned the microbiome in your introduction and you didn't talk about it. And then you said you moved institutions 5 different times in 5 different institutions and you see differences or you want to control for that phenotype. So what have you done on the microbiome side to convince yourself that you're
So I have not done anything on the microbiome side. Fortunately, because Craig Franklin was an early part of this project, helping me, actually, he gave me my first IL-10 knockout mice and kept them out there at Discovery Ridge, where the colony has gone to and from a few times. And the best advice he ever gave me was to save poop from all these mice. And so we have done nothing with it, but if anyone wants to, I have a lot of feces in the freezer. And we could look at microbiome drift, which I'm sure because of the moves, I'm sure we've had a lot.
Just a quick question. So you are using the collagenase to deplete the macrophage. I believe you use the IP injection, right?
Yes.
So I think that will remove systemically all the macrophage from the model, not only the perivascular.
Correct.
So how could we exclude the unspecific influence from all macrophage removal?
Yes, it's like I said, it's a real sledgehammer, and we did validate. We looked at splenic macrophages with flow cytometry, and they're wiped out as well. And these mice are, if you let them go, they're sick. They won't survive, particularly the IBD group. So this is a major macrophage depletion in the whole body.
Okay, and last question.
Erika, I'm just wondering about the adventitia. You talked about a lot of adventitia. Didn't talk about anything on the fibroblast resident, because in the large vessel, we know the macrophage creates adventitial inflammation. Do you think is there any role for the adventitial inflammation in your model with the macrophages?
I mean, I think fibroblasts could certainly be playing an important role. We have not labeled for them or counted them in any rigorous way. So I think it's absolutely possible that there's changes and that they're relevant, but we have not looked at that.
Okay, so before we thank Dr. Boerman, I want to make sure that I announce that please sign on next month on April the 13th. Dr. Kehoe Lee from Kaffner will be giving an overview of swine models for neurodegenerative disorders and diseases. But with that, thank you for your time and attention, and let's thank Dr. Boerman for a great talk.